Thanks all for being here. I'm Krissy, student in the Oceanography department at UH Manoa. Oh, sorry. I'm a graduate student at University of Hawaii Manoa, and I'm going to talk to you today about microbial source tracking, which we'll talk more about here in just a moment. The way that this is going to work out is I will talk to you first about microorganisms in the environment, and we'll talk a little bit about where they come from and where they're going. I'll then talk to you about a project that has been going on for the past couple of years on the Big Island. We'll talk a little bit about the sampling methods that we use for that project. And then this is a project which is in progress. So we'll talk about what our findings are so far, and then what the future directions of that project are going to be. So bacteria are all around us, and they are found in every different environment. And in this particular case, the bacteria in this picture are represented as having different shapes and different colors. Of course, these bacteria are not to scale. Bacteria are microorganisms that we can't see with our naked eyes. So in this picture, sorry, I forgot how the laser works. Oh, there we go. So in this picture, we have different shapes of bacteria. They have different colors. We find some of them in the terrestrial environment, but we also find them in the marine environment down here in the water. As animals, we have microbes that are associated with us. So we have microbes on our skin, but we also have a whole community of microbes in our guts. And the microbes in our guts help us to process the food that we eat and access the nutrients in our food. And what that then means is that when we rid ourselves of waste, shed along with that waste. And depending on how the waste is managed, those gut bacteria can find themselves in environments that we wouldn't typically find them. So in this case, we're finding gut bacteria in the marine environment, and this can lead to things like beach closures. So there's a couple of different ways that bacteria can get into the coastal environment. Like we saw on the previous slide, it could be through storm events or runoff. And what we're seeing in, what we see in this picture is this is a house that has a septic system. And if that septic system is not properly maintained, then we can get seeping of gut bacteria into the groundwater, which can then find itself on the coral reef. Now, in addition to beach closures, this can have environmental effects, because essentially what we're talking about is manure. So where we find gut bacteria, we also find nutrients. So if you think about this in terms of manure, in the terrestrial environment, we put manure on our plants and it helps them grow big and strong. But the nutrients that comes along with that manure can be used by ocean plants as well, right? So in this case, we're looking at a coral reef, which is now becoming an algal-dominated reef, because this started out as a coral reef, but now it's shifting to being dominated by algae, because the algae are better at getting that nutrients faster than the corals are, okay? But we should keep in mind that just because we find nutrients does not always mean that we're gonna find gut bacteria there. Nutrients could come from gut bacteria, but it could also just be regular old Miracle-Gro fertilizer, or we could get nutrients that already exist in the environment from maybe some sort of animal that's already present in that environment. Because it could be all of these different sources, it becomes very important to be able to determine where the nutrients are coming from and being able to identify what those sources of pollution could be. Okay, so I'm going to shift gears a little bit and talk to you about this project that we've been working on in a small residential community called Pu'ako. It's on the Kona side of the Big Island. And over the past 40 or so years, Pu'ako has experienced a decline in live coral cover. So in about the late 70s, there was about a 75% live coral cover. But this graph is taken from a report that came out in 2012, and it showed that in 2010, 2012, we really only had about a 30 to 35% live coral cover in this area. So the community of Pu'ako is very concerned with this and enlisted the help of folks at UH and also of the Nature Conservancy in order to try to determine what might be going on in this area and why we're seeing these kinds of declines. Let's see. So researchers then did a couple of different coral surveys, and what they found were growth anomalies and areas of algal overgrowth. So in this first column over here, we see coral growth anomalies. And what those are deformities in the coral skeleton that can actually have a physiological impact on the coral animal. In the middle column here, we see areas where algae actually grew on top of the corals. And as I was saying before, those algae can outcompete for nutrients or other resources in the area, and it causes the coral tissue underneath the algae to actually die. So these could be some of the reasons that we're seeing declines in this area. When the researchers graphed this and looked at the other influences that might be contributing to these types of anomalies, they found that where there were areas of fresh water, we found higher amounts of coral growth anomalies and also more dead corals. All right, so that then makes us wonder, what is coming in with this fresh water that could be causing these corals to be getting sick? So here's where microbes come into the picture. Microbial source tracking uses microorganisms in order to identify sources of pollution. And traditionally, we do what are called plate counts, which look like this. This is a method that's used by the EPA currently in order to determine human health risk. And what it does is identify what we call fecal indicator bacteria. These are bacteria that tell us that maybe fecal matter is in the area, but it doesn't allow us to determine the source of that fecal matter. Also, some of the bacteria that are used could potentially already be present in the environment. And so it doesn't necessarily mean that it's coming from fecal matter. But there's new methods that use DNA. And these DNA methods allow us to use a larger number of bacteria or a number of different types of bacteria. These methods are really specific. They're so specific, in fact, that we can tell the difference between gut bacteria that come from humans versus those that come from animals. And they allow us to get results faster. So in order to illustrate how these things work, let's go back to the beach and visit our scientist there. So she's going to go ahead and collect a water sample. And for the traditional plate count method, what she'll do is then take that water sample. She will put it onto this gridded filter. This allows her to collect all of the different bacteria that might be in the water. She then can put that filter onto a nutrient plate. And this then allows the bacteria to grow up. And her bacteria of interest are going to change color to blue. And then she can count the blue colonies, and that will tell her how many bacteria that potentially came from fecal matter exist in this environment. Okay, so that's the traditional method. We have a new method, which again, we would collect a water sample. We would get all the DNA out of that water sample. And then we would use different molecular methods. In this case, we're going to be looking at this magnifying glass, and it's going to show us the different bacteria in there. Now, if you remember, I said that these are very specific methods, and so this can allow us to identify all of the different bacteria from a given group. So in this case, we were looking for all of these rod-shaped bacteria. We can use then even more specific DNA sequences that allow us to identify just a subset of these. So it might be, these guys are supposed to be circling these orange rod shapes here. So it would require that it's rod shaped and that it's orange in color. Okay, so these methods have actually been used on the mainland and also in other countries in order to determine sources of nutrients and also to identify human sewage as opposed to other animals we might find fecal matter from. Okay, so we used actually both of these methods in our Pu'oko project. And what we found is that in areas where we had freshwater input, we also had higher numbers of bacteria. What this tells us is that in the case of the plate counting method, where we have freshwater, we have high bacteria, and it's probably coming from some sort of fecal matter. With our DNA method, we again find that with freshwater, we have a higher number of bacteria, but in this case, we were able to use the DNA marker to find gut bacteria specifically. So now we want to use a more specific method that allows us to look at human gut bacteria. And we don't actually find the same trend. And in fact, we still didn't find the same trend that I don't have like a nice pretty graph to show you. So we have to think about why this might be. Okay, so what's up with that? Gut microbes in this area might not be from humans, right? Like they could be from cows, like we saw in the earlier picture. But here's the thing. Along with this method, some other groups did some what they call dye tracer studies, where they took a dye and they put it into a wastewater system in this area, and then they looked at the coast margin to see if the dye showed up there, and if so, how long it took to get there. They found that it took between 6 hours and 3 days to show up on the coastline here. So we're pretty sure that what we're dealing with is human gut bacteria. So then what it could mean is that our method might either be too specific, and what that would look like is, again, if we come down to our little magnifying glass and we're trying to identify all of our orange rod-shaped bacteria, maybe we're identifying some of them, but not all of them, right? So like this guy got left out. The other problem could also be that maybe the bacteria that we're interested in are not the ones that we're looking for. So maybe we should be targeting these purple bacteria instead of the orange ones. So interestingly, a number of research articles recently have shown us that our gut microbial communities can be different. And the reasons that they're different are because of our diet, because of our geographic location that we grew up, and also because of our ethnic heritage. So where we're at right now with this project is the DNA targets that we currently use were developed on the mainland for use in the mainland. But we already know that Hawaii is unique for a lot of reasons, right? So we have a different diet out here. We have a number of international tourists that visit us. So potentially maybe the microbes that we're looking for aren't the ones that we actually have. So the next step in this is to take a closer look at the DNA sequences and determine whether or not we should be using different targets in order to look for our bacteria of interest, or if maybe our targets are simply just too specific. So with that, I would like to thank the co-authors of this study and also partners and our different funding sources, and we'll see what happens.
Okay, I'm kind of the odd one out here today. I'm from the economics department, so we're switching gears completely. The title of our project is Predicting Hawaii Water Demand Under Climate Change. Doing this project with Michael Roberts, Kim Burnett, Chris Water from the economics department, and Danielle Spirandelli from the Department of Urban and Regional Planning. And then I'm the lowly PhD student doing all of the number crunching. So the motive. Yep.
Is the climate change the global climate change or just?
It'll be focusing on Hawaii, and I'll talk about that in a little bit. So the motivation for this project is how will climate change affect supply and demand for water? So, and there are a number of ongoing studies that are looking at the supply. So what they're doing is looking at different models projecting overall increases in precipitation due to increased evaporation from climate change. But that's on a global scale. In a more micro scale, some areas will be drier, some areas will be a lot wetter. One thing that hasn't been looked at so much is the demand side. So how are consumers going to respond to increases in temperature, changes in precipitation, and how is that going to affect their water consumption? So that can affect consumption in many ways. Agriculture is a big one, landscaping, and air conditioning. You can imagine how all of these things are going to change when temperature changes. And this is going to depend on precipitation, temperature, prevailing climatic conditions. So consumers are also going to be responding. Demand is just as important to study as the supply. Why are we studying on Oahu? Several reasons. There's large climate uncertainty for climate-related factors. So there's uncertainty with how trade winds are going to be affected, which is going to affect how precipitation is occurring on the island. and we rely heavily on precipitation to recharge our aquifers, which is where we source our fresh water from for drinking and everything else. Limited research on demand response to climate. So like I said, lots of people are studying how precipitation patterns are going to be affected, how we're going to be looking at different changes in resupply of our aquifers, but no one's really looking at the demand side, the consumer side. Current water supply and demand are precariously balanced. So current estimates say that we are pretty close to our maximum sustainable yield. So if we start increasing our consumption of water a lot, we're not going to be drawing the water sustainably from our aquifers, and eventually we'll run into seawater and we'll have to start desalinating that, and that's really expensive and good to avoid. And the increased demand due to increases in populations and different things like that is going to also increase our need for water, and that's just going to confound with these other effects. Widely varying microclimate conditions within a single water district. So all of Oahu is under Board of Water Supply, so we all have the same pricing systems based off of the type of premise we are, basically. So all single family homes are under a single pricing system, but there's a lot of variation in the types of microclimates within the island. So Manoa is a lot different than Kapolei, for instance. So one that's really quite wet and a little cooler, the other is really dry, really hot. So there's a lot of variation that we can study to get at these effects that drives differences in demand. Okay, so this looks kind of scary, but it's not that bad. What we're doing really is, oops, that's the wrong button. So a simple way to model this would be to say that the consumer's monthly use in water is some function of characteristics and prices, and then maybe the average temperature over that billing period. And that's a really fast way to look at it. It's really easy computationally. But when we're only looking at averages, we're going to be washing out all of the variation within a billing period. So what we're going to be doing instead is looking at the full distribution of temperatures and other weather variables within a given month. So just to make that a little bit simpler, let's say that water consumption depends on temperature and temperature squared. What we can do then, simplify or expand rather that regression equation we had on the last slide, and we're weighting basically each degree by the amount of time that the building has been exposed to that temperature. So instead of just looking at the average temperature of the building throughout a billing period, we're going to weight it by how much time has been at each degree. So the technical thing is we're getting at nonlinear effects. So it's just a linear effect when we're only looking at averages. You get a lot more detail basically by breaking down the days and hours within a billing period. And that really helps with the explanatory power of the model. Oops. So for the data, climate and weather data, we have baselines. This is a really fine baseline. It's the 250 meter resolution of the entire island. And it gives us an average temperature for every hour of the month. So it gives us something like, for instance, average temperature at noon in January for a particular point in the island. So those are the baselines. We downloaded hourly weather data, so the actual temperatures for 4 weather stations across the island. And those are 4 airports. And I'll show it on a map in a minute. But we got data from 1990 through 2015 on an hourly basis.
When you say 32 stations, what's that?
So right now we only have the 4 stations. So we have hourly weather data for 4 stations. Currently, we gained access to an even better data set that has 32 stations. So we're currently improving our weather estimates.
Where are the 32 stations?
They're all over the island. Yep. Just this entire project is just Oahu. So right now we have 4 stations on Oahu. We're expanding that to 32 stations, which is going to really help at getting detailed weather data for specific points on the island. After some struggling with Board of Water Supply, they gave us 3 years of data so far, and we're trying to get even a longer history to get more billing data. And this is really detailed data. So we have the amount of water they consumed for a particular building. So we have the monthly billing data of every building on the island, basically. So we're combining this really detailed water consumption data with the really detailed weather data to get at the consumers' responses to temperature, precipitation, things like that. So this is how we constructed the fine scale weather data. I don't want to go into it too much, but basically, so the 4 blue boxes represent the locations of the weather stations we have data for. So we have hourly weather data from 1990 through 2015 for these 4 stations. And this isn't to scale, but we have a 250 meter resolution baseline. So just average temperatures for every other point on the island.
What is anomalies?
So what we do for any given point on the island is we can calculate the estimated hourly temperature at a particular point in time using these anomalies, or what is what I'm going to call it. So we have hourly actual temperatures, for instance, for these 4 stations, and we know their baseline values.
That's an anomaly.
So the anomaly would be the ratio of the actual to the baseline value. So it would say maybe our baseline temperature for a particular point is 20 degrees, and then the actual hourly reading was 25 degrees. So that would mean that we have a 20% higher than average temperature at that point in time.
You probably could have called it deviation. I mean, you picked an anomaly for this particular function, definition. You probably could have called it deviation.
Yeah, right. So it's like a deviation from the mean. I just didn't want to make it sound like that it was a subtraction type thing. So we calculated the anomaly or deviation, whatever you would like to call it, for every hour for these 4 stations. And then we used those anomalies to calculate anomalies for every other point using inverse distance weighting. So we could estimate the actual hourly temperature at every point in the island using the baselines and just hourly temperature data from 4 stations. And with our 32 stations that are scattered even more throughout the island, you can imagine that our estimates are going to be improved a lot once that's finished. So that's how we constructed the hourly weather data. Now we have to look, so this is what I've come up with so far, actually. Let's see. Just play a little bit of it. So at the top, we have the hour and date. And as it goes through, you can see the differences in temperature that we've estimated throughout time. And we're going to match this with the billing data from Board of Water Supply to use in that regression that we showed. Okay. So this is the distribution of the types of premises that we have. Almost all of them, 92%, are single-family homes, and they're pretty evenly distributed throughout the island. The rest of the 8%, there's lots of commercial buildings, low-rise residential buildings, and then everything from agricultural to industrial, government composes the rest. And this is percent of total consumption. So single-family homes drink about 40% of all the water that we have data for, and then low-rise residential, commercial are some other big consumers. I've found the relative consumption. So this is, you can think of this like gallons per building for each type of premise. So things like golf courses, they use a lot of water. High-rise residential buildings use a lot of water. Hotels use a lot of water. And for some reason, federal government uses a lot of water as well. That's the biggest consumer of water. And those were mostly military bases. So now we've matched the premises with the temperatures. So this is just for residential as an example. So January through December, in the summer months, it's really hot. High-rise residential is the red, and then on the other extreme is the purple single-family homes. Most high-rise residential buildings are located in town, which is on average warmer than the single-family homes, which are distributed more evenly throughout the island. So now we can compare water consumption to temperature. So I know I said that using the averages got rid of a lot of detail, but this is just preliminary work, so it's what I'm working with so far. So on the horizontal axis, we have average billing temperature and average consumption of water on the vertical axis. So as it's warm, as you have a warmer billing period, you're going to be consuming more water. And for now, this is just single family homes, since those are the biggest group that we had. What also matters is where you're located. So this is average temperature for a house over the 3 years that we have data for. So people that are living in really warm areas drink more water than people that are living in relatively cool areas. So what I wanted to show was that not only does the day-to-day temperature matter, which is this blue set of box plots here, but what also matters is where you're located, how hot your microclimate is, I guess. And we did the same thing for precipitation as well. So when you have a rainier billing period, people tend to drink less water. And that's where we're at so far. So right now we've constructed our preliminary weather data set and we've matched it with the billing data that we have. We're trying to get more billing data so that we can improve our results in addition to improving our weather data using the additional stations. So once we get all of that, we can develop our model. We can actually use the regression that I showed in the beginning. And that's going to let us understand better the effects of block pricing structures. We have price data, and more importantly, variation in weather, climate, and building variations. Once we have those regressions finished, we want to expand on it by using projections of the climate. So we're going to get data for projected changes in precipitation and temperature to see how climate change is going to affect the amount of water that the residents, commercial buildings, industries are consuming water on Oahu. Right, so different climate scenarios, different development scenarios. So how is development in a certain area going to affect the water that's needed in that area? And pricing policies is one other thing that we might look at if we have time. But that's it.
Aloha, everybody. I'm Eduardo Guimaraes, and today I'll share a little bit about my project. Rapid response application of a qPCR-based test for enterococci as a rapid beach management tool in Hawaii. It's a project done by Water Resources Research Center from University of Hawaii at Manoa and Institute of Marine Sciences from University of North Carolina, Chapel Hill. So before we start, I think you guys might be asking what is enterococci. It's pretty hard to spell. That's bacteria, and we use that as an indicator of human sewage in the water in Hawaii. So let's get started. Water quality, I mean, who likes good water here? I mean, I like it. I was in Honolulu Bay today and it was awesome. What do we care? So what do we care about good water? What do we care about not inputting sewage in the environment? So human sewage in the environment especially can have several implications. So an environmental implication of that is eutrophication. So that means we have very little and dissolved oxygen. So fish are actually not able to breathe when we input a lot of human sewage in the environment. Human sewage can also have a lot of pathogens, so people get sick from it. We can contract diarrhea, for example. And climate change is supposed to increase and intensify the number of storms. And that's going to increase also the amount of bacteria we're inputting in our environment. Not only environmental and human health issues, also economical issues. It's calculated that about 3.5 million people get sick due to swimming in bad water just in the US. And only the Orange County itself loses about 21 to 51 million dollars per year. Just, yeah.
What is that? Eutrophication.
Oh, yeah, eutrophication is an effect in water. Is what happens when we input sewage, human sewage in the environment. So human sewage has a lot of nutrients, and that develops, as Krissy said, a lot of algae. And when the algae dies, it consumes oxygen, and fish are able to breathe without oxygen, right? And then they die. It's very sad. So yeah, 21 to 51 million per year only in the Orange County. That's because people don't go to the beach and they don't go to the restaurants and they go to hospitals and have to spend money, right? Water quality is even more important in Hawaii. We all know that we receive annually millions of tourists, and they all expect to find a very safe environment and very clean, pristine beaches, and they don't expect to get sick, right? So water quality is extremely important for us. This looks great for swimming. It's in Kaneohe Bay. However, we're not just able to know if the water is proper for swimming just by looking at it. In this case, Kaneohe Bay is not proper for swimming. Waikiki has had several sewage spills, mainly due to overflow of the system. So it's very important for us to know when the water is not good for swimming, because we run into several problems every year. How do we know? How do we measure water quality in Hawaii? How do we know if water is bad for swimming? So we follow Environmental Protection Agency recreational water criteria, which has standards for enterococci, as I mentioned, it is the bacteria, and C. perfringens, which is another bacteria for sewage indicator. And the standards are 130 colony forming units per 100 milliliters for enterococci, and 50 colony forming units per 100 milliliters of C. perfringens. So one colony forming unit is 1 dot in this picture on the right. And we measure those standards by using culture incubation methods, which is also the same thing Krissy mentioned. And is the thing on the right here, this picture on the right. And the problem with that is that these are very old method and it's not very efficient. It takes up to 48 hours from sample collection to getting the results from the method. Oh, what happens? Oh, oops. Okay, sorry. So what that means, sorry. That means that when we see warnings like that, like this, the ocean water is not good for swimming and you should not get in. What means is that ocean water quality was bad yesterday and you should not have swam. And water quality might be okay today. Please check our website tomorrow. So there's a delay in from sample collection to results, and that's not good. We want to know exactly if we can get in the water and if it's safe for locals and for tourists. So how does Hawaii fit with rapid methods? Hawaii is extremely well suited. So as I mentioned, we get about 8 million tourists a year, and they mostly concentrate in areas like Waikiki and La Moana. And those two beaches, they are very easy to reach from the city, from the university, from the lab. So we can go collect the sample, and we believe we can post the results within 3 hours. So people are able to know in a daily basis how is the water in that moment. And that's the machine, that's the qPCR. That's how we do the close to real time sample and posting. So this stands for quantitative polymerase chain reaction, and that allows the rapid detection of and quantification of any DNA from any organism. So what we do, we put the samples in this tray here on the left, and some chemistry as well. And after we have this tray ready, we put this in this machine, and it goes through some chemistry, some different temperatures, and we get the result within 3 hours, 3 hours. This method is already being used in other states. So panel in California, in Doheny State Beach, is actually in a restaurant, I think. And it says here, Friday, July 2nd, 2010, last update at 11:30 a.m. and says the water quality is bad. So the public is able to know if they can swim. And in this case, let's say in Hawaii, we will only be able to know if the water quality is bad only in July 4th, because there's this 48-hour delay from sample collection to results. So that's bad. You know, you don't wanna, you wanna know if the water is good for you to swim, if it's safe, right? As I mentioned, we have to develop a human assay. We are currently using intercocci assay. Why we can use intercocci? Intercocci is known to grow in tropical environments, in outside host environments. So they grow outside our guts. So if we find high quantities of intercocci in the environment, they can come from just leaf debris or from humid soil. That can lead to unnecessary beach closure, because if it doesn't come from the humans, it's not dangerous for us. Humans, they have the biggest amount of pathogens in our sewage. So we can see here in this picture on the right that birds have salmonella, ruminants have E. coli, for example, and humans have all of those and some other dangerous pathogens. That's why we want to know and develop a human-specific assay, because human sewage is the most dangerous for us and for the environment in general. The assay we want to validate in Hawaii, it was already validated on the mainland. So is this assay here, developed by those EPA, and they are better than the previous human-specific assay in terms of specificity and detection at low concentrations. I know Krissy mentioned about specificity. This is where we can find this bacteria. So in the case of the old assay, we were able to find the bacteria in humans, dogs, and mongoose. We don't want that. We want to find this bacteria only in humans, because we want to close the beach only when human sewage, the dangerous human sewage, is present. So we expect high specificity. We only find the fecal indicator bacteria in humans. We also want to validate our assay through high sensitivity. This is, we find these fecal indicator bacteria in all of us, all of the humans. And high practicality. This is, we want the concentrations of this fecal indicator bacteria to be higher or just as high as intercocci. Just to wrap it up, the main objectives of this study. So we are trying to implement a rapid qPCR method for intercocci, as well as human-associated bacteroides, as a sewage tracer in Hawaii coastal waters. We expect this assay to have high specificity, sensitivity, and practicality. We also want to evaluate the feasibility of the daily use of assay. So we expect to collect the samples by 7 a.m. and post the results by 10 a.m. to the public. We expect to promote the education of stakeholder groups through workshops like this one, for example. These groups can be Surf Rider, City and County of Honolulu, Department of Health. And hopefully after this study, we're gonna have a more reliable method to provide safety for locals and also tourists that come here, and we can inform better about the water quality of Hawaii. Just like to thank you, Sea Grant, for the funding and for the support for this project, and also my mentor, Marakirs, for the help.
So we're gonna have.
Nationality are you, and how long have you been here?
I'm Brazilian, and I've been since 2013.
When?
Since 2013? No, I was doing undergrad before, and now I'm doing grad school. Yeah. Okay.
So we're gonna have the graduate students stand up, and if you have any questions for them, or any questions for our topics.
I have one question. Remember you showed water consumption W? W was representing water consumption?
Yes.
Remember it was a function of the temperature? My question is, why was it also a function of the temperature squared?
That was just an example. We don't know exactly what function we use. We have to do some calibration to figure out what's going to fit the data the best. I just wanted to quickly go through that because it was simple. And we know that it's probably going to be some sort of non-linear relationship. So it's not just going to say, if you go from 10 degrees to 20 degrees, maybe you drink twice as much water, but 20 to 30 degrees might have a different relationship. So that's what the non-linear.
Now, did you ever consider maybe there's a temperature cube function in here?
Oh yeah, there certainly could be. We just don't. That was just an example. I mean, the equation was already pretty long. I didn't want to add anything else to it.
But you include a more practical thing. You studied the water consumption and the baseline and all. Did you come up with at least a rough idea of when we would exceed our consumption would exceed what we have? I mean, it's true, depending on what the temperature might be, but whatever model you use, did you come up with a critical year, max?
Not yet, but so that's the plan. Right now we just have historical data. So we're specifically looking at the relationship between water consumption and temperature. We haven't implemented the future models yet, and there's lots of different future models. So some are very conservative estimates of what's going to happen, some are pretty liberal. So depending on which one actually occurs will determine some sort of, I guess, critical year, like you say. But other studies say that we're pretty close to our, it's called the maximum sustainable yield. So right now, we're kind of at an equilibrium, but if we increase our consumption anymore or precipitation starts dropping off considerably, we're going to start running out of water.
I have a question either for Krissy or Lawrence, or not necessarily for your talks, but I was wondering, it's two-part. One is there like a standard sort of like time to sample, like at least X number of days after rainfall or whatever, just to keep everything standardized? And then the second part is, I've heard that there's actually very little water quality testing in Hawaii, and I was wondering, because I've heard that percentages are only tested once a year. Sounds like maybe they test quite a few more often to even using this old method, how often they test it. You guys know, like, do you have a field or?
So you said the best time once we implement the methods is to collect the samples by morning, because then people are going to come later on, right?
Yeah.
But regarding rainfall, I'm not sure, but I would expect, depending on the time of the year, if it's very dry and then there's a big rainstorm,
Right.
we expect the first flush effect, right, to make the water very polluted.
Yeah.
But I'm not sure if that affects or not how the Department of Health collect their samples or not. I'm not sure.
So for us, ours is like a little bit more just kind of research oriented and less monitoring oriented. So for us, because we're trying to find groundwater signatures, we would probably want to test when the tide is out so that we can get that bigger pulse of groundwater, right, and capture it better that way. But as far as like how regularly monitoring agencies are testing around here specifically, I don't really have a feel.
I would say it's very little, and that's not good, because we know that Hawaii, for example, is not very safe, and we know that there are the system is not very safe in Hawaii. There's some leakage, and we all like surfing, in Baila Moana, and that's close to Hawaii and stuff. So it's not very safe, and we hope that once we have the method, we're able to help that.
So like he's not regularly tested?
I wouldn't say so. It is actually. It is.
Oh, it is.
It is.
Okay.
My mentor is giving me some tips.
Okay.
Yeah. Then we hope also, it's expensive, and we hope that the hotels can fund all of this testing because they are the biggest interest, There's a study that says that when people get sick, they actually don't return spend their money here into the hotel. So, and the hotels get so much money, and hopefully they could fund this whole structure. Yeah.
Talking about the funds, do we have any federal funding to help this program continue?
So for our research, I'm getting some funding from Sea Grant also in order to do the DNA side of the work, to be able to look at the sequences that are already there and then determine what the best sequences to use here might be. But other than that, I don't know that, I'm not getting any from.
No, I think just Sea Grant.
Stephen?
Yes. Yeah.